Okay. We left off the other day. We were talking about things that go on inside of a muscle cell. Okay. We mentioned the idea of mitochondria, right? So we're going to pick up here. This is kind of where we left off. And you guys don't have to be responsible for the individual internal structures or where various metabolic processes are going to take place in a mitochondria for this test. We're going to get to it. Maybe we will for this test. We will get to this, okay? But you don't have to know it today, all right? This is a mitochondria. Here's a cartoon of what it looks like, right? Kind of looks like a little, like a bean or something. And then inside, there's going to be all of these extra folds and things that we call cristae that are going to provide us with surface area. And here's an electron, a micrograph of a mitochondria, so you can see all the different membranes and the folds and what all of this looks like, okay? Here is, right, the real kind of action sites in the mitochondria, and it's going to be where aerobic metabolism takes place, okay? For most of you guys, you're sitting here right now, your mitochondria are supplying the vast majority of the ATP that you need to stay alive, stay awake, to have your heart beat, to breathe, to be able to type, whatever it is that's going on right now. Okay, so we'll get into where exactly does the Krebs cycle take place, why are the concentration gradients across the different membranes the way that they are, and why is that going to be important as we move along. Okay. All right. If it will advance. Okay. I know that I mentioned this as well, so I just wanted to highlight it again. We talked about myonuclei and satellite cells. We talked about that muscle fibers are giant in the cell world. And because they're giant, they have to have many nuclei. Okay. And so here is a muscle fiber under a microscope. The little things that look like maggots or whatever on there are nuclei or they're satellite cells. I told you guys under a microscope, in a lot of instances, it's hard for us to know which ones are which. I know these look like they're in the middle, but they're actually on the outside. It's just, if you think three-dimensionally, the fiber is facing you, and these nuclei and satellite cells are just on that outer portion of the membrane that's facing you guys. Okay? The big thing with the nuclei and with the satellite cells, especially the nuclei, though, is they control protein synthesis. They control growth and repair in skeletal muscle, right? And so we need lots of these to supply our fiber, to keep it healthy, and to let it grow, okay? Satellite cells are stem cells. This is a drawing, in essence, of where the satellite cells are going to be located, and they're going to be in a very similar locale, right, to the nuclei. The only way that you can tell them apart is really under very high-powered microscopy, that they're going to be out here on the edge, and they're sort of encased in a double membrane, whereas the nuclei aren't. But these are really important. We'll cover those stem cells more so when we go along. Okay. All right. We feeling okay about that, right? Whole muscle, fascicle, fiber. We've talked about some of the structures in the fiber. Okay. Now we're going to start drilling down a little bit farther. Okay. And the big part of going down farther and smaller in this way is as we get progressively smaller, this is where the actual force production, okay, is going to come from inside a muscle. So if the muscle has one job, it's to generate force, but that force is going to come from the interaction of actin and myosin proteins together. Those actin and myosin proteins are going to be organized into a sarcomere, which looks like this, and then we're going to stack sarcomeres end to end to make a myofibril, and then we bundle up a bunch of myofibrils together inside of our muscle fiber. Okay. The entire point of this organizational structure is get the most actin and myosin in as small of an area as we possibly can, so we can maximize our ability to generate force. Okay. Maximize our ability to generate force. That's what is going to go on here. So I'll walk you guys through a little bit of this. We don't spend much time about myofibrils, other than here's what they are, they exist. The real action, the real kind of functional piece though, is going to be the sarcomeres that get stacked end to end to end to end and make up a myofiber. So here's one sarcomere, okay? The edge of the sarcomere here and here is called the Z line, okay? So the borders of a sarcomere is called a Z line. I do want you all to know that, okay? Other than that, if you look at stuff in the book, they'll talk about different areas inside the sarcomere. There's an A band and an I band and an H zone and an M area and all of these things. Don't concern yourself with all of that. Don't concern yourself with that, okay? What you need to know about sarcomere is that in between the Z lines, okay, we are going to stack up and pack together in 3 dimensions actin filaments, which are these kind of gold and yellow looking filaments here, and myosin filaments, which are the that kind of an orangey red looking things here. Okay. And so actin filaments, actin is a smaller molecular weight molecule. They're little spheres. We stick the spheres next to each other. We take 2 strands of them and we wind them together, and you get what's called an actin filament, or it's sometimes called a thin filament. Okay, a thin filament. I'll show you all an electron micrograph in the next slide, and you will see that the reason they call it thin is because it's smaller and it doesn't stain as dark. Okay. Those are going to be here. Myosin filaments are what we call thick filaments. Myosin is a much larger protein than actin. Myosin has its own unique structure. Okay, if you look down here at this, it has these head molecules and a tail region. I'll show you guys some kind of zoomed in cartoons of what this looks like, because these individual structures in the myosin are going to be important. Okay, there's an enzyme on myosin. There's a couple of other regulatory sites on myosin that help us generate force, that determine how quickly it generates force, and that determine how much ATP it uses when it breaks itself back off. Okay. Differences in myosin are going to be primarily what determines differences in fiber, muscle fiber types, which we'll talk about in a few slides. Okay, so thick myosin filament, thin actin filament, and the idea behind the sarcomere is to pack these things together. Okay, pack them all together so that we can have as much myosin adjacent to or near actin as possible, so that these myosin heads can attach to actin, form what we call a cross bridge, generate force, and drag them up, sort of drag these things towards the middle and shorten and cause a muscle contraction. Okay, so in that way, the sarcomere really is the smallest functional unit of a muscle. Okay, force production is going to come from here. These things slide, they pull on the Z line. The Z line of one sarcomere pulls on an adjacent one, pulls on an adjacent one, pulls on connective tissue, pulls on the fiber, and eventually is going to be what's going to make movement and force transmission onto the tendon. Okay, here's an electron micrograph of sarcomere, or many sarcomeres. Okay, and so what you see as you look through on so many of these things, here to here, Z lines, and then these stripes in the middle are going to be the darker bands are myosin, the slightly lighter bands, which look kind of gray. You almost have to back up a little farther. Y'all can see them probably better than I can. Are going to be actin, and you can see from all of this how highly organized, how tightly packed, okay, all of this is going to be. All right. This is what skeletal muscle and cardiac muscle looks like. You look at sarcomeres in smooth muscle that like lines your digestive system and things, and it is a huge hot mess. It does not look like this. So that's one of the kind of key things about skeletal muscle. Okay. Further, what I'm trying to illustrate with this cartoon, okay, is I really want to try to get you guys to think about muscles and think about how things work in our body in 3 dimensions as much as possible. Okay, as much as possible in 3 dimensions. And so here's how you normally see a sarcomere. Okay, what we have taken here is we have taken, right, this sarcomere, but all of the sarcomeres, like sort of in a myofibril, and we've turned it out to face you guys. So on this one, you're looking down into, okay, down into the myofibril. And so what you see then is here we have this, and in 3 dimensions, we have thick filaments in pink, thin filaments in blue. So there's this hexagon arrangement of 6 actin molecules that surround one myosin molecule. Again, this allows us to pack as much actin and myosin into a particular area to maximize, when we need it, our ability to bind those 2 things together to generate force. Okay, so this is trying to show the same thing, right? You can see kind of the hexagonal shape around all of this. Each one of these things would represent kind of where the myosin heads are going to all come off at these 60-degree angles to be able to bind onto this. Okay, does that make sense? Okay. I'm going to show you a little bit about a myosin, then we're going to talk a little bit about some regulatory proteins on actin, and then we'll get into fiber type. All right. I'm going to tell you about this, and I do not want you to fuss over what the hell myromycin is and light versus heavy and all of that. I show you this because what I really want to do is just make you aware of a couple of things that are happening on the myosin molecule itself. Okay, so thick myosin filaments, right, is going to be sort of one myosin molecule that looks like this, and then it's a bunch of these things, sort of the tails all sort of fit together. All right, and where the hinges are, where the head is very, very important, but a little beyond the scope. That's a little bit more kind of master's level stuff. All right, so a thick filament is made up of this sort of, the tail region is made of something called light myromycin, and then we have this thing here called heavy myromycin, which is up at the front. But then within the head and part of all of this, there's also something called the myosin heavy chain and the myosin light chain, and you've all gone cross-eyed and changed your major because this is completely ridiculous, okay? But what I want you to appreciate about the head region is, in the head region of myosin, there are multiple different kinds of little proteins and molecules that are going to be there. Okay. The myosin heavy chain is important because this is the thing that we grab with an antibody and stain to figure out, is it a fast twitch fiber? Is it a slow twitch fiber? Okay. We will talk about looking at myosin MHC. The MHC stands for myosin heavy chain. So different fiber types have slightly different variations of these kind of open circle molecules that are going to be around the outside of the head. Then the other thing to keep in mind is there are these light chain molecules on the inside, which they're important, but they're again beyond our scope. But I want you to see right here, okay, right at the end, there's a binding site for actin. This is the piece of myosin that grabs onto the actin. And then right next to it is an enzyme called an ATPase, or the myosin ATPase, okay? The reason you use energy during exercise is because of this enzyme right here, okay? This enzyme right here. How quickly this enzyme can split ATP into ADP is arguably the single biggest predictor of how fast each one of you guys can run, of how high you can jump, okay? because the speed of this enzyme differs based upon muscle fiber type. Okay. The fast twitch fibers that allow someone to be a good sprinter, to be an explosive jumper, and those kinds of things, it uses a huge amount of ATP and it splits it very quickly. Whereas in the slow twitch fiber, it splits it slower and it's more metabolically efficient. So it's this region on this one protein, okay, that plays a huge role in metabolism during exercise, in actual performance characteristics of people during exercise. Okay, that's why I show you guys all of this, because we're going to talk about it in an abstract sense. We're going to talk about, okay, you're a fast twitcher, you're a slow twitcher, you have this kind of profile metabolically, or this or something else, but it all comes back to this enzyme, right? Okay. That enzyme right there. Okay. Now, there's a couple of other things on the actin filament that we haven't talked about that are going to be really, really important. Okay. And we call them regulatory proteins. Okay. Regulatory proteins. Anybody having a cramp right now? Anybody cramping? No? Anybody having an unwanted muscle contraction? That is, Stacey, y'all are? Okay. A little bit of something. For the rest of all, you know why you're not having an unwanted muscle contraction? Because of a molecule called troponin and a molecule called tropomyosin. Okay. I use lots of analogies about kids, right? When you have little kids around the house, you got to put the little plastic things and all the little power outlets, right? The plug-ins, right? To child-proof them. Basically, troponin and tropomyosin are the child-proofing on your actin molecules. Okay. Tropomyosin is this rope-like structure that lays on the actin molecule, and in its normal state, at rest, it covers up the binding site between actin and myosin. Okay. It's the plug that we put in the wall that covers up the outlet. Troponin is another protein that sits at kind of regular intervals along the actin, sits on there and connects to the tropomyosin, and it waits. Okay. Troponin waits, and it will bind calcium. All that calcium coming out of the sarcoplasmic reticulum that we talked about on Wednesday, it's released for a reason, and that reason is to bind to troponin. It binds to troponin, it changes its shape, and it pulls the plug out of the wall. And it opens up the binding site. As soon as the binding site is open, here comes myosin, and it wants to grab onto actin. Okay. So the reason our default state on actin, therefore our default state in skeletal muscle, is no contraction. The default state is keep actin and myosin from being able to bind together. This way, okay, rather than having to turn something off, right? When you're getting contractions, it costs us energy. That's not great, right? Potentially. So the default is no contraction. When we want a contraction, then we'll move this stuff. Then we can get it, and then we'll shut it right back off again. Okay. So that's why these are called regulatory. They regulate the actin binding site. Okay. Y'all with me so far? Do we need, I didn't tell you guys on Monday. One, when I get some names and learn y'all's names, I'll just call on people. If I feel like y'all are not paying adequate attention or doing things, we'll just get up and do squats until somebody answers. Okay. Yeah, we'll just squat. We'll wall sit, we'll squat. It doesn't really matter. Okay. Y'all will do it until I'm tired. And then maybe that will prompt everybody to answer. Okay. So we've talked about the individual filaments. We've talked about the individual proteins and how they're regulated. Okay. Here now is what all of this looks like. All of our skeletal muscle structure, all of our organization is going to come down to our ability to maximize the potential to do exactly what is on this slide. And that's form a cross bridge. When actin and myosin bind together, We call that, when they are bound, a cross bridge. Okay, cross bridge. We must form cross bridges to get force. Force production at any point is directly related to the number of cross bridges that are engaged. Why is a bigger muscle likely stronger than a smaller muscle? There's more actin and myosin, there's more ability to form cross bridges. It's not exactly that simple, but in the very, very big picture, it is. Okay, so here we are at rest. Okay, tropomyosin sits on the actin binding site. Thick filaments very close. This myosin molecule is, it's just waiting. All you have to do is just open the door, and it's coming in, it's going to bind. Okay, we release calcium, it binds to troponin. We'll talk about next week all the upstream steps of what's going to happen to get the calcium to come out. But we release calcium, it binds to troponin. Okay, we move tropomyosin. Now these purple binding sites are open. Myosin binds. Boom. Cross bridge, force. Okay, force. This is what's going to happen. This right here is what lets us move. It's what lets us exercise. What lets us breathe. This is happening in your heart right now. You never have to think about it. Okay, this is how muscles function on their most basic level. Okay. Once it binds, that myosin head grabs on, and it's like a tug of war. Okay, it's going to grab and it's going to try to pull that actin molecule towards the middle. It's going to try to make our sarcomeres short. Okay, that is called the sliding filament theory of a muscle contraction. The myosin pulls on the actins. Okay, it doesn't pull on every actin unless it's a maximal contraction, but it pulls on the actin. The actins are all connected together at the Z disc. And so as one gets pulled, it drags everything else along. Okay, and as that sarcomere comes together and shortens, then eventually we'll get enough sarcomere shortening that it will pull on a tendon, and that will pull on a bone, and we'll start to get an actual muscle contraction. Okay, you have millions, hundreds of millions of sarcomeres in a muscle. Okay, if I told you I want you to contract one sarcomere in your bicep right now, one, can't do that, right? That's too fine of a thing. All right, but you can imagine a scenario whereby as I start off, I start with one and I work up through hundreds and thousands, and I eventually get enough that it will allow me to sort of move. Okay, when the myosin head tilts and pulls the actin, we call that a power stroke. Okay, a power stroke. Once it binds and it tries to initiate a power stroke, we have done about half of something that's called the cross bridge cycle. Okay, because here's the problem that we run into with all of this. Okay, imagine I'm a myosin molecule. This is actin. I grab it and I pull. That has moved like 2 micrometers. Okay, not nearly enough to actually get movement in our bodies. And I've pulled it and it's here. Well, now what? Okay, I need to be able to go again, right? And so in that instance, what the myosin molecule has to do is let go of the actin. I got to step back over here so I've got some room again, grab on and pull again. Okay, that process from grab, pull, break, reset, grab, pull, break, reset, that's called the cross bridge cycle. Okay, that's called the cross bridge cycle. And it's illustrated here. We're going to start at the top. Okay, this is a resting sarcomere, right? No binding, we're just waiting. We're at rest. It's very, very important that you guys understand where there is ATP, where there is inorganic phosphate, and where there is ADP in this process, because what happens right here is why running uphill is hard. It's why walking downhill is easier. This is where, okay, the primary place where energy use during exercise occurs is in this cross bridge cycle. So we've got to, it is absolutely key to understand and pay attention. In a resting muscle, all myosin heads have a molecule of ADP and a molecule of inorganic phosphate that have been split apart attached to them. Okay? They're here, they're resting, they're waiting. Okay? We get calcium, we open up the binding site, it binds and forms a cross bridge. Okay? Here. As that happens, we release inorganic phosphate. Okay? As the inorganic phosphate is released, that's the thing that initiates the tilt or the power stroke in the myosin head. So it bends and pulls. As it does that, it then releases ADP. Okay? It releases ADP. This is a muscle contraction, or this is what leads to a muscle contraction. In what is maybe the most conceptually backwards thing we will talk about in the entire class. We have generated force, we've initiated movement with this, we have yet to use any energy. Okay? We've used no energy yet. All we have done is just let go of something. We've used no energy. Okay? And we're like, what? Yeah, no energy. It doesn't take energy or use energy to initiate a power stroke. Okay? What happens after the power stroke, though, is where we use energy. ATP comes in, binds, and that tells the myosin to let go of the actin. Then that ATP, with the myosin ATPase, and then that ATP gets broken down into ADP and inorganic phosphate, which stay on, and we start the whole thing over. So technically, you don't use energy to generate force. You use energy to get rid of force. You use energy to get myosin to let go of actin so we can start the whole thing over again. Over and over and over again. Okay? So when we get to metabolism, we will talk about contractions and exercise and things that cost us more energy. They're going to cost us more energy because we go through this cycle more often. The more we go through this cycle of grab, tilt, let go, right? Back, grab, tilt, let go, recock. The more we go through all of this, that's where we use the energy. So let's do critical thinking really quickly. Very, very broad sense, and we'll cover more of this next week, okay? If I told you guys, right, we took a straw poll. My wife's big on straw poll. Usually involves when I'm like, people don't do this, or she's upset with me about something. She's like, let's take a straw poll of other people and what they think. They should, of course, do the dishes or those things. So, right? Use more energy going upstairs or going downstairs? Going up a flight of stairs or down? Up, okay, yeah, sure, right? Is gravity the same regardless? Do you weigh the same regardless because of gravity? So it should, in theory, take the same force to move you up as it does in the opposite direction to keep you from falling down when you're coming downstairs. And yet, you use like half as much energy going downstairs, going down a flight of stairs as you do going up. So the question is, based upon just what I've told you, why? A very reasonable argument, but when we look at the force production, I mean, if you're going with gravity so much, you just fall, right? I can go with gravity, I just fall down. I'm using force to counteract gravity to stay upright. So the actual difference in force production is actually very, very small in those things, okay? Based just on this.
You're going down with something catching, you're not having to contract muscles as much, I guess.
Okay.
So you're not having to have your mouse heads receive ATP to track on.
That's right. So you're 75% correct. Okay. You're contracting just as much you're getting just as many cross bridges, because remember, force is related to cross bridges. When you go down, you're doing an eccentric contraction for the most part. And when you do eccentric contractions, you actually don't have to break them apart. They just grab on and they get pulled and they stretch a little bit. And so the necessity of doing the cross bridge cycle, as you pointed out, is way less going downhill or going down a flight of stairs. And so it's much more metabolically taxing to go up than it is to go down. And you all have that experience, right? If I said you want to run up this hill, you want to jog down this hill, everyone's gonna be like, down, I want to go down, right? Like this is the thing. Okay. So we'll keep that in mind as we go on. But that's how we're going to transfer what's happening on a protein level. Okay, not just a cellular level, but on an individual protein level to what you see, what you feel, what you experience during a bout of exercise. It all is going to come back to this, right? To this. I have a question.
Could you repeat what you said about eccentric, about how there's not that break apart for it? Just make sure I heard that correctly.
So, and we will get into this when we talk about eccentric contractions next week and versus concentric. When you do an eccentric contraction, okay, I am lowering my phone down. So I'm doing an eccentric contraction with my bicep and whatever the other elbow flexor muscles are, right? As I come down like this, okay, I am forming cross bridges to generate force that's just a tiny bit less force than whatever my phone weighs, right? If I do this and I match whatever it weighs, it doesn't move. If I want it to go down, I want gravity to win, but it's very controlled. It's just a tiny bit less than what it weighs, and I lower it down. I form a cross bridge. I get that force, but then as I let gravity start to win, all that's happening in a lot of those sarcomeres is just, it grabs on, and because of the stretchiness in the sarcomere and the connective tissue, it just gets stretched out a little bit.
Okay. Part of why it leads to like with tempo, eccentricity and with optimal loading for that, is that part of why it helps the hypertrophy process?
That's a much more complicated question. There is something magic about eccentric contractions for sure. And we will get into a little bit of that, but that's probably beyond where we want to be today. But keep that in mind. We will come back to it, I promise. Okay. Any other questions before we do 10 quick minutes on fiber types and then we get you guys out of here so I can at least be on the syllabus? You guys will note by the end, I was like a whole week of like to be determined. There's no fucking way that's going to be the case. Like we'll be behind on things in the midst of this. Okay. All right. Here is a thing called the muscle biopsy. I used to in class talk about the history of exercise physiology. There's a nice chapter in the book if you care to look at it about all of this. But this procedure right here was one of the seminal advances in all of exercise physiology in the late 60s. Okay. We cut open your muscle. We jam this thing called a Bergstrom biopsy needle into your muscle. We apply suction. We pull muscle fibers into this little window. Then you slide a plunger down. This thing works like a guillotine and it cuts off a piece of muscle. We then pull everything out. Some poor grad student like I had to do when I was a PhD student pulls this out. You take this, you mount it. As you can see, it was like an eraser on a pencil. You mount it on a popsicle stick and then you freeze it in liquid nitrogen. And then you got to go put it in the world's smallest deli slicer and cut these little bitty thin slices off of it so you can look at the individual muscle fibers under a microscope. Okay. This is called a muscle biopsy. This was the first time we had the ability to look inside intact human skeletal muscle and see what all was going on, right? In animals, we just kill the animal, we take its entire muscle. I assume none of you guys are volunteering for that experiment, right? On those things. And so this is what led to all of this. And what we realized when we began to look at all of this is that not all muscle fibers are the same. They're not all the same. Okay. And that led to the advent or the discovery that they come in different fiber types. Okay. I know you guys have heard of fiber types before. You've heard of slow twitch and fast twitch, or what we'll call type 1 and type 2 and 2A and 2X and all these hybrid fibers, which we'll get into in the next slide. But at its most basic initially, it was like there's two kinds. Okay, there's two kinds. First initial way we did this in the late 60s was through something called histochemistry. You take the fibers, you bathe them in ATP and a couple of different stains, and they figured out that that myosin ATPase molecule on the myosin head broke down that ATP at different speeds. Okay. And so from doing this with histochemistry, we figured out there's two primary kinds, some that do it fast, some that do it slower. It's still very fast, but it's just slower than the fast. Okay. They were also able with histochemistry to look at metabolic enzymes, things in the Krebs cycle, things in glycolysis. Okay. Hexokinase is a glycolytic enzyme. SDH stands for succinate dehydrogenase. It's like the fourth step in the Krebs cycle. LDH is lactate dehydrogenase. It's when we make lactic acid. Or we could look at with histochemistry how much glycogen is in your muscle, how much glucose is in your muscle, how much lipid is in your muscle. So this was kind of the initial things. What we do now with them, who's taking a rapid COVID test? Probably everybody in here. Okay. If you've taken a rapid COVID test, they work through something called immunohistochemistry. Okay. There are antibodies that bind to things that are byproducts of the virus, right? You swab your nose, you get pieces of those virus on there, you put them in there, it runs down the column, and then it runs across those enzymes and it binds to them, those antibodies, it binds to them. The antibodies connect things that light up. That's how that works. We do this now, much more so than histochemistry. You take a biopsy, you grind it up. We have an antibody that's very specific to all of the different isoforms of myosin ATPase, of succinate dehydrogenase, all of the things that are going to go on there. We do this through what's called immunohistochemistry. Okay. Primarily with this, when we want a fiber type, we look at MHC or the differences in myosin heavy chain. Okay. The differences in myosin heavy chain. So, told you guys that in the 60s and in the early 70s, we based it on myosin ATPase. So there were fast muscles and there were slow muscles, right? Fast fibers and slow fibers. That's still a perfectly reasonable way to fiber type people. Okay. But what we have learned as our molecular biological techniques have gotten better, that when we look at the myosin heavy chain isoforms, we have slow twitch muscles, or what we call type 1. There's one kind of slow twitch fiber, one. But within fibers that have a fast myosin ATPase, they come in several different flavors. Okay. And so we call fast ATPase fibers type 2. But based upon their myosin heavy chain, we have 2A, 2X, 2B, and then there are what we call hybrid fibers that exist on a continuum in between all of these. 2A fibers are the most aerobically metabolically adapted type 2 fibers. Okay. They are fast with their ATPase, but they tend to have a lot of mitochondria, a lot of glycogen. Right? If you are any sort of an endurance athlete, basically all of your type 2 fibers, whatever you have will be 2X. 2Xs are even faster and they're wildly metabolically inefficient. Okay. But those tend to be the fastest fibers in humans are 2X. Some people have a little bit of 2B. 2B is really like an animal, like a cat and a rat and a mouse. Things that are even faster than us, right? Tend to have those that are going to be in there. And then we have these hybrids that exist in the midst of this. I'll say this now, okay, because it's really, really important. All of you have some genetically determined initial distribution of 1s versus 2s in your body. 80% of y'all are somewhere 60-40 one way or the other. Slow to fast or fast to slow. Okay. There's a little bit of variation among muscles in our body, like your diaphragm is super slow. For everybody, it's super slow. Okay. Postural muscles like your soleus, right? Your rector spinae, those kinds of things tend to be more slow. They have greater, if you're 60-40 in your quad, you might be 80-20, right? You're 60-40 slow twitch to fast twitch in your quad, you might be 80-20 in your soleus, okay? But in general, everybody has some initial starting range. You cannot change that. You cannot change it, okay? You are born to be a sprinter or you're not. I am one that was not. You were born to be a sprinter or you are not. Okay. We cannot switch slow twitch to fast switch. We cannot switch fast switch to slow twitch. I say that, that's not 100% accurate. If you have a spinal cord injury and you don't contract a muscle for 2 years, you will turn your type 1s into type 2s. But that's like the only extreme circumstance. Okay. Within type 2, you can convert among your type 2 fibers. So if you do basically any kind of physical activity, your 2X's become 2A's. If you do a lot of aerobic kind of training, you're going to get a lot of 2A's. They're going to have a lot of mitochondria in those 2A's. I can make every one of you a significantly better distance athlete. That's not a problem if you're willing to work. The range on all of that, because of the metabolic shifts in type 2 fibers, is pretty big. You're either a sprinter or you're not. Sorry, that's just the way that it is. You're fast or you're not. You're explosive and you can jump or you can't. Yes, we can increase your strength. We can change your form. We can do a little bit, right? We can do a little bit. But I'm never running under 10 seconds in the 100 meters. And that's because of fire. All right, we'll clean a couple of things up with that, but we'll move on to the nervous system side of things on Monday. Please look, finish your Connect assignment for today. There's another one for Monday. Please get those in. If you're having problems, let me know. All right?